Membrane potential. Electrical charges are fundamental to the proper functioning of all cells. Everything depends on electrical potential, from the heartbeat to the movement, the absorption of the sugar that we ingest, our thoughts, and up to the understanding of what I'm saying now. All of this depends on ion displacement. Before we start with the cell membrane, let's first see the electrochemical gradient on a simpler system. Solids diffuse from one compartment to another following the chemical gradient, that is the difference in concentration between the two compartments. The gradient must be considered individually for each chemical species that can cross the membrane. Although the two containers have the same concentration, 4 molecules each, they are imbalanced, and this is because there is a gradient for both yellow and green molecules. Balance is reached with the passage of a yellow molecule to the right and a green molecule to the left. But let's move the goalpost by adding charges to our molecules. Now, the displacement depends not only on the chemical gradient, but also on the electric gradient. In a very intuitive way, positive charges repel each other instead of staying grouped in the same container. A substantial difference is that the electrical gradient does not depend on the different types of molecules, but only on the overall charge. Whether it is a sodium ion, potassium ion, or cationic molecules, what matters is the difference of charges between one compartment and another. So, we solve first the electric gradient by moving a charge from the more numerous container and then the chemical gradient. In reality, gradients balance at the same time, and there is no priority order. But let's see one last example to understand how a cell membrane works. Compared to the previous one, the channel connecting the two containers is narrow, and not all chemical species can pass. When a chemical species cannot diffuse its gradient, it's not considered from the chemical point of view. Although the yellow molecules are ignored from the chemical gradient point of view, we'll still consider them for the electrical one. 3 reddish molecules, in fact, would have to go from the first to the second vessel to cancel the chemical gradient, but this would create a strong electrical gradient because many positive charges would repel them back. Even if there is still a chemical and electrical gradient, they act on the same molecules in an equal and opposite way, canceling each other. For each difference in concentration of an ion, there is an electric potential that can compensate for the chemical gradient. For those who are familiar with chemistry, this potential can be calculated with a Nernst equation. Now that these concepts are clear, let's move on to our cell membrane. As we have already pointed out, the membrane separates the intracellular compartment from the extracellular one. These two compartments do not have the same concentration of ions and molecules. To understand the potential, we'd better consider only the substances with charge and present in large amounts. The potassium ion is strongly concentrated inside and the sodium ion outside. As for the negative charges, we have chlorine mainly concentrated outside, and then inside the cell instead there are large anionic proteins. So let's leave out the negative charges that, except in some cases, are not fundamental to determine the membrane potential, and let's focus on the positive charges that will be the protagonists of this topic. The different concentrations of sodium and potassium are constantly maintained by pump, the sodium-potassium exchanger. By consuming ATP, the pump is able to carry 3 sodium ions outside and 2 potassium ions inside. 2 chemical gradients are created, but the 2 ions cannot diffuse freely across the membrane. However, the cell does have some permeability to ions, although low due to the presence of some always open ion channels. There are many more channels for potassium than for sodium, and the permeability of the membrane to potassium is therefore 50 to 100 times greater than that for sodium. Some of the potassium, therefore, leaves the cell according to its chemical gradient. In this way, the membrane loses positive charges and the inside of the cell becomes negative compared to the outside. Normally, the inside is at minus 70 MV relative to the outside. This value is actually significant and varies from about minus 60 to minus 90 MV depending on the cell type. What is important to understand is that this potential is maintained due to the constant activation of the sodium-potassium pump that creates a chemical gradient, and the slow release of the potassium ion according to the gradient determines the negativity of the membrane potential. If the sodium-potassium pump stopped working, the sodium and potassium will slowly spread from the two sides, canceling both the chemical and the electrical gradient. This potential that is constantly and actively maintained is called resting potential and represents the basal situation of all cells. There are particular ion channels that, in response to stimuli, can open or close. The closing of potassium channels, or more frequently the opening of sodium channels, determine an accumulation of positive charges inside the cell. In this way, the resting potential of -70 MV is raised, and this is because the internal environment is less negative. If it reaches -55 MV, then an important event is triggered: membrane depolarization. At that potential of -55 MV, voltage-controlled sodium channels open. Sodium, at this point, enters massively due to the large concentration difference. In this way, the membrane potential is not dissipated, but reversed. Now, there are more positive charges inside the cell than outside. For this reason, this phase is called depolarization. During depolarization, the potential rises to a maximum of +35 MV, after which the voltage-controlled sodium channels become inactivated, preventing further sodium entry into the cell. If sodium could exit freely, it would in fact rise to +65 MV. The reduction of membrane negativity, however, has not only opened the sodium channels, but also those of potassium that, however, by their nature are slower to open and take over with a certain delay. These channels let out potassium according to concentration gradient at a very high flow, much higher than that allowed by the channels constantly open in the resting phase. The release of positive charges represented by potassium restores the membrane potential, and these channels should close again. But as they were slow in opening, they are also slow in closing. So potassium leakage is such as to restore not only the membrane potential, but to make it even more negative than the -70 MV that we saw earlier. The membrane is therefore hyperpolarized. This is where the initial example vessels becomes useful. Potassium that is now practically free to flow according to different gradients spreads until it creates a potential difference such that it is pushed back even if there is still a concentration gradient. This balanced potential is near -90 MV, and this is why the cell polarizes at this stage. When the membrane potential returns negative, the sodium channel returns from its inactive conformation to its closed conformation. We'll speak about this difference in a moment. So the membrane is back with a negative potential, but this is not yet the resting situation. The electrical potential has returned more or less in place, and the chemical potential is still altered. In the cell, there are still large concentrations of sodium ions, while outside there is a concentration of potassium. The resting condition is restored by the slow passive spread of ions through the membrane and the constant functioning of the sodium-potassium pump that restores the chemical balance. So, let's go back to the voltage-controlled sodium channel that we've left behind. So this channel in resting condition when the membrane potential is more negative than -55 MV is in a closed conformation. It is called closed because a depolarizing stimulus that increases the membrane potential can open it and trigger depolarization due to the violent entry of sodium. If a depolarizing stimulus fails to reach -55 MV, the channel does not open and the stimulus is ignored. This is called the all or nothing law. As we've seen, between -55 MV and +35 MV, the channel is open. At higher potentials, it becomes inactive. Also, in this state, the channel does not allow sodium to pass, but unlike the closed state, a depolarizing stimulus is not able to open the channel. For this reason, it is impossible to superposition two depolarization waves, but it is necessary that the membrane is repolarized before a second one starts. Let's review schematically all of these electrical events that collectively take the name of action potential. In the resting condition, the membrane potential is maintained at -70 MV by the sodium-potassium pump and the slow flow of potassium ions across the membrane. A stimulus that may be represented by a neurotransmitter, a change in temperature, or mechanical stretching induces a slight increase in the membrane potential. If this depolarizing stimulus is not able to reach -55 MV, it is ignored and the resting potential is slowly restored. If instead it reaches the critical threshold, the voltage-dependent sodium channels are activated and a huge influx of sodium enters the cell and the membrane potential reverses. Reaching +35 MV, the sodium channels are inactivated and the membrane is refractory to any other stimulus. For this reason, this state is called absolute refractory period. At this point, however, potassium channels have opened and they restore the membrane potential by letting potassium out. Because of their slow response to changes in potential, potassium continues to escape up to -90 MV, hyperpolarizing the cell. At this stage, in theory, the cell can initiate a new action potential following a stimulus. Now that the membrane is hyperpolarized, it is difficult for a stimulus to reach -55 MV unless it is very intense. For this reason, this state is called relative refractory period. Finally, the restoration of the correct intra and extracellular concentrations is achieved through the activity of the sodium-potassium pump. The action potential is a very fast event that lasts 2 to 3 milliseconds. However, this is the potential of neurons and skeletal muscle. There are also other types of action potentials, such as that of cardiac muscle that involve the entry of other ions, in this case calcium, which allow the state of absolute refractory period to be maintained for hundreds of milliseconds. This is critical to ensure that the heart has a proper contraction. We gotta point out that the electrical chemical events that we've described are relative to the near membrane environment, and compared to the enormous size of the cell and extracellular environments, changes in ion concentrations are irrelevant. The changes in concentrations are relative only to the small area where depolarization occurs, but do not significantly change the total concentrations of sodium and potassium. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.